to prepare our structure to input into Rosetta? Well, I just have a couple of things listed up here. Structures from the protein data bank are not perfect, right? Everyone, I'm not sure if everyone may have opened up a protein, looked at it, and there's obviously a clash, right? It's not perfect. You can't always trust the protein data bank. If you were to put that into Rosetta, it may fail. Rosetta may not work. The worst thing that could happen, though, is that Rosetta doesn't complain about the protein, and it runs the protocol all the way through, and you look at your results and they look fine, but the actual input structure was incorrect, and it goes all the way down to the end, and you could just spend a long time messing with a protein that doesn't actually work. So garbage in, garbage out. It does it silently. You have no way of telling. Another benefit to looking at your structure beforehand and cleaning it up is you spend less time correcting mistakes in the protein in your protocol. So for example, if you're doing relax, which is a minimization, if there's a clash in your protein, right, and you keep on using that as the input structure, it's going to spend the same amount of time trying to resolve that clash. When if you fixed it once, you would never have to spend that same amount of time to fix it over and over again. There's less noise in your sample, and you get overall better scores. So what do we do? This was asked to the Rosetta community a couple of months back, and basically, lots of people replied to this, and they said, we clean the structure, we repack the structure, we relax the structure. I'm going to tell you how the Mileway lab does it, and this is actually what people thought was the best answer. I've included in the scripts directory, there's this thing called cleanpdb.py, and what it does is I'll go into details later, but it cleans your protein, and once it's clean, you will run relax, and I'll show you what all these things mean. So cleanpdb.py. This script was created ages ago in the Baker lab, and it's been modified ever since, and then we got our hands on it, and we modified it for this conference so that you guys can use it. What it does is it will remove all the het atom records, all the ligand files, all the waters, all that stuff. It changes the names of phosphorylated residues, sulfated residues, different types of residues that you would see. It puts it in Rosetta nomenclature, so Rosetta understands it. For example, for phosphorylated serine, it will rename it as SER, but it will keep the phosphorylation on there. It's just now that Rosetta, when it reads the protein, it knows that it's a phosphorylated residue and will run normally. It renumbers the protein starting at number 1. This is very important. I can't tell you how important this is, because whenever you are using Rosetta, if, for example, your protein is a dual-chained protein and it goes from 1 to 50 and then starts back at 1 to 50, what Rosetta will do is read it as 1 through 100. It doesn't read it separately as 1 through 50, 1 through 50. And so whenever you then look at it, you can spend a lot of time just looking at the output of Rosetta and being like, what? These numbers don't correspond. You could be looking at the wrong residue. So what this script will do is renumber all the residues so that they start at 1. It will not build missing density. So if your crystal structure has missing density, it will not build those in. What you need to do is run loop modeling, which Liz is going to go over. It also doesn't add hydrogens. Rosetta will do that for you. What it does not do is very important. It doesn't build missing coordinates, and it doesn't solve all your problems. There might still be problems in the crystal structure that you need to go in and look at and fix manually or think about. You're going to by doing Python space cleanpdb.py-h, and it will give you very specific directions. I said this earlier. What is relax? What relax does is it refines the structure using Monte Carlo movements of the backbone. So it will, it's basically a minimization of Monte Carlo movements. And you can use it to generate multiple conformations to start your structure protocol out with. Yes, you look like you have a question.
What kind of minimizations does Rosetta do? If it is minimization or differentiated minimization?
It's a Monte Carlo. Move the backbone. Is this good? Yes, then drive it down to
So it is not iterative.
it's not what?
It's not iterative. The energy is not that close down.
It does. Because it does a gradient-based minimization after it changes the backbone. It does gradient-based minimization. Will this be accepted or rejected? Monte Carlo. If it's accepted, it accepts the pose. If it's rejected, it does another move and it.
Is the same way as folding?
I'm not sure.
Is minimization? The minimization.
When you do minimization and folding, is that your question?
After folding.
After folding, yes. Then it's the same protocol. And it's actually, it cycles through, it repacks the structure, minimizes, does some backbone movements and stuff. It's documented fairly well on the documentation. And so this is very important. This is something that came up from just talking to you guys whenever you are, when you were sending me emails and asking me questions. When you're doing design, ligand docking, protein docking, you need to do some different things. It's not okay to just have one model and dock a ligand into it. You really want multiple models, and you want to dock into multiple backbones. And to do this, you would you run relax, and you do instruct 100, and this will output 100 relaxed models. Take the top 10% by energy. This is maybe 1%. You know, I would just give you one model. Top 10% by energy, and use those as starting points to dock into. It's very important that you do that, and this is the reason why. Here's an energy landscape, and in ND, right, you can maybe start right here, and it'll traverse until it gets into the low energy minimum. Sometimes it won't make it over here. I'm just showing this as an example. In Rosetta, you do the same starting point. You start right here, this high energy, but you need to do random sampling, right? You're doing Monte Carlo moves, and it's accepting, rejecting. The more starting models, the more starting positions that you have, the better coverage you get of this energy landscape. And then if you have enough, you'll actually get in the right spot, whereas MD would never get into this spot. Then you would take this model and then put it into MD and further drive it down.